Hello everyone. Today we are going to discuss about bioremediation. So let's get started. What is bioremediation? Actually, the term bioremediation is made up of two words. Bio means biological and remediation means solution. In simple term, bioremediation is the biological process in which we are finding out some solution to the problem occurring in that particular area. It may be soil, water, or any environmental area or any particular site. So we'll see the definition of bioremediation. Bioremediation is the use of microorganisms, bacteria and fungi to degrade the environmental contaminants into less toxic form. In this process, we use the microorganism to degrade the contaminant site or area to convert it into less toxic form. It actually purifies the area. The microorganisms may be indigenous to a contaminated area, means a microorganism present in that particular area or originally belongs to that area, or they may be isolated from contaminated site. The process of bioremediation can be monitored indirectly by measuring oxygen reduction potential or redox in soil and groundwater together with pH, temperature, oxygen content, electron acceptor, donor concentration and concentration of breakdown products. Example, carbon dioxide. In situ bioremediation, it involves treatment of contaminated material at the site. It means degradation is carried out at a contaminated site. Advantage, low cost method. Disadvantage, time consuming. There are three types of bioremediation: biosparging, biostimulation, bioaugmentation. So we'll see biosparging. Injection of air under pressure below the water table to increase groundwater oxygen concentration and enhance the rate of degradation. In this method, groundwater oxygen concentration is increased by injecting the air under pressure to enhance the rate of degradation. Biostimulation. Bioremediation can be carried out by bacteria that is naturally present in environment or adding nutrients. This process is called biostimulation, involving the addition of nutrient, water, electron donor or acceptor that stimulates microbial growth. In biostimulation, microbial growth is stimulated by adding nutrients, water or electron donor or acceptor. Bioaugmentation. Microorganisms are imported to a contaminated site to enhance biodegradation process. In this process, microorganisms are added to a contaminated site to speed up the rate of reaction. Ex situ bioremediation. It involves the removal of contaminated material to be treated elsewhere. It means contaminated material are transferred to the another site for treatment. Advantage, faster method. Technical complications, it involves land farming, biopile, bioreactors and composting. What are the similarities between bioaugmentation and biostimulation? Bioaugmentation and biostimulation are two sustainable treatment methods to clean polluted areas. Biostimulation can be enhanced by bioaugmentation. In both processes, microorganisms are utilized. Both methods are biological methods. These methods do not result in toxic byproducts and are not harmful, unlike chemical methods. Both methods has received much attention nowadays due to their potential and sustainability. They are promising and long-term solution for the degradation of contaminated soil and water. Moreover, they are cost-effective methods unlike chemical methods. So basically the major difference between bioaugmentation and biostimulation is that in biostimulation the microorganisms are stimulated. In bioaugmentation, microorganisms are already present in contaminated site. Both the methods are cost effective unlike chemical methods, and byproducts which are formed are not harmful at all because natural organisms are involved in this process. Problems associated with bioaugmentation. There are abiotic and biotic stresses causing the death of introduced organisms. In addition, from indigenous organisms almost always face competition from indigenous organisms for limited nutrients. It can be difficult to deliver the inoculant to the desired location. Advantages and disadvantages of bioremediation. So we'll see advantages. Useful for complete destruction of wide variety of contaminants. The complete destruction of target pollutants is possible. Less expensive, environment friendly. Disadvantages. It is limited to those compounds that are biodegradable. Not all compounds are susceptible to rapid and complete degradation. Biological processes are often highly specific. It often takes long time than other treatment process. So we'll see factors affecting bioremediation: nutrients availability, moisture content, pH and temperature. These all are factors that affects the bioremediation process. Factors affecting bioremediation: nutrients availability, moisture content, pH and temperature are some factors that affects the bioremediation process. First we'll see nutrients availability. Inorganic nutrients such as nitrogen and phosphorus are necessary for microbial activity and cell growth. It has been shown that treating petroleum contaminated soil with nitrogen can increase cell growth rate, decrease the microbial lag phase, help to maintain microbial populations at high activity levels, and increase the rate of hydrocarbon degradation. Moisture content: all soil microorganisms require moisture for cell growth and function. Availability of water affects circulation after water and soluble nutrients into and out of microorganism cell. However, excess moisture such as in saturated soil is undesirable because it reduces amount of available oxygen for aerobic respiration. So we'll see pH. The pH of the solution or water determines the nature of the species present in it, as it can cause the change in chemical composition due to the basic and acidic nature of the site. Generally, the pH range from 5 to 9 is considered optimum for the working of various microbial colonies. As biological reactions are all pH sensitive, so existence and functioning of all enzymes are highly pH sensitive. Temperature: it is one of the important factors that defines the moisture content and chemical composition of the site. Generally, the temperature between 20 and 40 is considered good for the efficient performance of microbes. Fluctuation in above parameters can affect the bioremediation process. Thank you.